All right, welcome everyone. Today we're going to tackle that incredible journey from fertilization all the way to the formation of the placenta. We'll connect all the high-yield dots, turning what can seem like a really complex sequence of events into a clear, logical story that you can confidently recall for your exams.
So, have you ever felt like early embryology is just a blur of events happening way too fast? A lot of students find it hard to connect everything. Our goal today is to slow it down, break it down logically, step by step, and really build a solid conceptual framework you can rely on.
So here's our game plan. We're going to walk through this journey chronologically in 4 key stages. We'll start with fertilization, that initial spark. Then we'll follow the embryo as it finds a home during implantation. And finally, we'll do a deep dive into the placenta itself, its structure, circulation, and its many, many functions. Okay, let's dive right in at the very beginning with that first, absolutely critical event, fertilization. This is the moment it all begins, when a sperm fuses with an ovum. So what's the whole point of fertilization? Well, it's got 2 really big jobs to do. The first is pretty obvious, right? It's to kickstart the development of a brand new embryo. But the second is just as important, and it's all about the genetics. It's to restore the normal diploid number of chromosomes. You get 23 from the sperm, 23 from the ovum, and boom, you have the full set of 46 in that new cell, the zygote. Now, this image here is just perfect for seeing that first week's journey. Take a look at where fertilization happens. It's way out of the ampulla, which is the widest part of the fallopian tube. And that location is absolutely key. Why? Because it gives the new zygote a few days to travel, all while it's dividing and developing into a blastocyst, getting itself ready for the big event, implantation in the uterus. Okay, this is one of those high-yield slides you'll definitely want to remember for your exams. Let's run through the key dates. About 30 hours in, we're at the 2-cell stage. By day 4, this ball of cells, now called a morula, enters the uterine cavity. Around day 6, implantation kicks off. And by day 10 or 11, we see the very beginnings of uteral placental circulation. By day 21, the villi are fully mature. Okay, so our little blastocyst has made it to the uterus, but its journey is far from over. Now comes the next huge challenge, implantation. This is the whole process of embedding itself into that welcoming, receptive uterine wall. You know, implantation isn't just one thing. It's actually a 4-step process, a carefully choreographed dance. It starts with what we call apposition, which is just some initial loose contact. Then it gets more serious with adhesion, a firm attachment. That's followed by penetration, where it breaks through the surface layer, and finally invasion, where the blastocyst burrows deep down into the underlying tissue. And wow, this image really brings that invasion step to life, doesn't it? The key player here, the one doing all the work, is the syncytial trophoblast. That's the outer layer of the embryo. Think of it as this highly invasive front line. It actively eats away at the maternal tissue, including blood vessels, and that's what creates these little pools, or lacunae. These spaces then fill up with maternal blood, establishing that very first, crucial connection. Now, while all this invasion is happening, the endometrium isn't just sitting there passively. It goes through this amazing transformation and becomes what we call the decidua. This new, specialized lining is packed with nutrients like glycogen and lipids, which is perfect for feeding the early embryo. And, very importantly, it also helps to control just how far that trophoblast can invade. This diagram breaks down the decidua into its 3 key regions. You've got the capsularis, which covers the embryo like a little cap. You have the parietalus, which lines the rest of the uterus. But the one you absolutely need to remember is the decidua basilis. Why? Because that's the layer right underneath the embryo, and it's going to form the maternal side of the placenta. It's the foundation. All right, so the stage is set. Let's now zoom in on the star of the show that develops from this whole feto-maternal interaction, the placenta itself. We'll look at it from the outside in. So when we look at a full-term placenta, it's like it has 2 totally different personalities. On one side, the fetal side, it's smooth, shiny, almost glistening. That's because it's covered by the amnion, and you can see those big umbilical vessels branching out. But flip it over, and the maternal side is the complete opposite. It's rough, reddish, and broken up into these 15 to 20 lobes called cotyledons. This was the side stuck to the uterus. Okay, let's get out our microscopes. The real workhorses of the placenta are these things called chorionic villi.
They're the functional units. And they don't just appear fully formed, they mature in 3 key stages. Primary villi are just simple projections. Then they become secondary villi when a core of mesoderm grows into them. And the final, most important stage is the tertiary villus, and that's when fetal capillaries develop inside that core. Now we have circulation.
Okay, pay close attention here because this is probably one of the most important concepts to grasp. For the placenta to work properly, the maternal spiral arteries have to be completely remodeled. They transform into these wide, low-resistance vessels. This allows a huge volume of blood to flow in, but at a very low pressure, which is crucial so it doesn't damage the delicate villi. So you get this gentle fountain of blood that fills the space, bathes the villi, allows for exchange, and then drains away. It's a truly genius, high-flow, low-pressure system. So we've seen what the placenta looks like. But what does it actually do? Well, it's so much more than just a simple filter. It's this incredibly dynamic, selective barrier that's controlling every single thing that passes between the mother and the fetus.
The placenta is like a super smart gatekeeper. It uses different methods to transport different things. Small stuff like oxygen and CO2 just diffuses across, no problem. Glucose, which is the baby's main fuel, needs a little help. It uses facilitated diffusion. But then for the really important building blocks like amino acids and iron, the placenta has to actively pump them across. And for big molecules like the mother's antibodies, it uses a process called endocytosis to literally gulp them up and pass them to the baby.
All right, here's something that often trips people up. The placental barrier is not the same throughout the pregnancy. It actually changes, and it changes a lot. Early on, it's pretty thick, with several layers. But as pregnancy goes on and the fetus's demands just skyrocket, that barrier has to become way more efficient, so it thins out dramatically, by more than 10 times. Layers fuse and disappear until you have what's called the vasculosyncytial membrane, where fetal blood and maternal blood are incredibly close. It's a brilliant adaptation to maximize exchange when it matters most. So, when you step back and look at the whole picture, the placenta is just this incredible multitasking organ. I mean, think about it. It's the baby's lungs, its gut, its kidneys, and its main hormone factory, all rolled into one. It's a barrier, a protector, and it even makes sure the mother's immune system doesn't reject the fetus. It does it all. Okay, let's do a quick fire round of the absolute must-know high-yield points for your exams. If you remember nothing else, remember these. 1. Fertilization happens in the ampulla. 2. Implantation starts on day 6. 3. The decidua basalis makes the maternal side of the placenta. 4. Maternal blood bathes the villi and the intervillous space. And 5. The placental barrier gets thinner as pregnancy progresses. Got it? Good. So, what's the bottom line? This whole incredibly timed journey creates a very special type of placenta called a hemochorial placenta. Let's break that word down. Hemo means blood, and chorial refers to the fetal villi. It literally means the fetal tissues are bathed directly in maternal blood. And this incredibly efficient design is the ultimate lifeline for the growing fetus. And that wraps up our deep dive. But I want to leave you with a clinical question to chew on. We've just seen how complex and perfectly timed this all is. So what happens when it goes wrong? Think about things like ectopic pregnancies or preeclampsia. Understanding this fundamental biology is the absolute key to understanding the pathology.